In this video, I'm going to be going over the metabolism poster that we did throughout this course, which focuses on how the major metabolic pathways in biochemistry are interconnected rather than functioning as isolated systems. This poster follows how carbohydrates, lipids, amino acids, and nucleotides are metabolized and how they ultimately contribute to ATP production, biosynthesis, and cellular homeostasis. The pathways show how metabolites move between glycolysis, gluconeogenesis, the citric acid cycle, lipid metabolism, amino acid metabolism, oxidative phosphorylation, and nucleotide synthesis. Throughout this presentation, I'll also point out important regulatory enzymes, energy-producing steps, and key intermediates that connect multiple pathways together. So first, I'll start with glycolysis, which is one of the central pathways of metabolism. Glycolysis occurs in the cytosol and converts one glucose molecule into two pyruvate molecules. The pathway begins when glucose is phosphorylated by hexokinase to form glucose 6-phosphate. This traps glucose inside the cell. Glucose 6-phosphate is then converted into fructose 6-phosphate by phosphoglucose isomerase. One of the most important regulatory steps occurs when phosphofructokinase 1 converts fructose 6-phosphate into fructose 1,6-bisphosphate. This is considered the commitment step of glycolysis and is highly regulated by the energy status of the cell. Fructose 1,6-bisphosphate is split by aldolase into glyceraldehyde 3-phosphate and dihydroxyacetone acetone phosphate. Triose phosphate isomerase allows these molecules to convert. Later in glycolysis, glyceraldehyde 3-phosphate dehydrogenase produces NADH, and ATP is generated through the substrate level phosphorylation at phosphoglycerate kinase and pyruvate kinase. The final product is pyruvate, which becomes a major branching point in metabolism. I also included galactose metabolism through the Lilor pathway. Dietary lactose is broken down into glucose and galactose. Galactose is phosphorylated by galactokinase to form galactose 1-phosphate. Through a series of reactions involving UDP galactose and UDP glucose, it is converted into glucose 1-phosphate and eventually glucose 6-phosphate so it can enter glycolysis. I also showed fructose metabolism, which fructose can be converted into fructose 1-phosphate and eventually enter glycolysis through glyceraldehyde and dihydroxyacetone phosphate intermediates. Next is gluconeogenesis, which is essentially the synthesis of glucose during fasting states. Although gluconeogenesis shares many reversible glycolytic enzymes, the irreversible glycolytic step must be bypassed with different enzymes. Pyruvate is first converted into oxaloacetate by pyruvate carboxylase. Oxaloacetate is then converted into phosphoenolpyruvate by phosphophenolpyruvate carboxykinase. Another important bypass enzyme is fructose 1,6-bisphosphate, which removes a phosphate group from fructose 1,6-bisphosphate. Finally, glucose 6-phosphatase converts glucose 6-phosphate into free glucose. I also included how lactate and glycerol can feed into gluconeogenesis. Glycerol from triglyceride breakdown can be converted into dihydroxyacetone phosphate, which ultimately enters the pathway. Once pyruvate enters the mitochondria, it is converted into acetyl-CoA by the pyruvate dehydrogenase complex. This reaction is extremely important because it links glycolysis to the citric acid cycle. The pyruvate dehydrogenase complex requires several cofactors including TPP, lipoic acid, FAD, NAD+, and coenzyme A. Acetyl-CoA then combines 2-oxaloacetate to form citrate through citrate synthesis, beginning the citric acid cycle. The citric acid cycle completely oxidizes acetyl-CoA and carbon dioxide while producing reduced electron carriers. Several important energy-producing reactions occur here. Isocitrate dehydrogenase produces NADH and carbon dioxide. Alpha-ketoglutarate dehydrogenase also produces NADH and carbon dioxide. Succinyl-CoA synthetase produces GTP through substrate-level phosphorylation. And succinate dehydrogenase produces FADH2, and malate dehydrogenase produces NADH. So ultimately, acetyl-CoA is then converted into 3 NADH, FADH2, GTP, and 2 CO2s. These reduced electron carriers are then used in oxidative phosphorylation to generate ATP. The electron transport chain occurs in the inner mitochondria membrane and is where most ATP production occurs. NADH donates electrons to complex 1 while FADH2 enters through complex 2. Electrons move through coenzyme Q, complex 3, cytochrome C, and finally complex 4, where oxygen acts as the final acceptor. At complex 4, oxygen combines with electrons and protons to form water. As electrons move through the chain, proton pumping occurs at complex 1, 3, and 4, creating a proton gradient in the inner membrane space. The proton gradient then powers ATP synthase through chemoosmosis, which is ADP plus pi ultimately converts into ATP. As protons move back then into the mitochondrial matrix through ATP synthase, the released energy drives ATP production. Also, lipid metabolism and beta oxidation were included. So fatty acids are first activated to fatty acyl-CoA and then repeatedly shortened through beta oxidation. Each cycle produces acetyl-CoA, NADH, and FADH2. So fatty acid acyl-CoA then, like I said, produces acetyl-CoA plus NADH and NADH2. The acetyl-CoA generated can enter the citric acid cycle or be used for ketone body synthesis during fasting. Also, cholesterol synthesis is included, which is a major regulatory step in the conversion of HMG-CoA into malevolate by HMG-CoA reductase. This is the rate-limiting step of cholesterol biosynthesis and is also the target of statin medications. Another part of this poster included amino acid metabolism and nitrogen disposal. Transammination reaction allow amino acid groups to be transferred between molecules using PLP-dependent aminotransferases. So, for example, pyruvate can be converted into alanine and oxalate can be converted into aspartate. Glutamate plays a central role in nitrogen metabolism and can release ammonia through glutamate dehydrogenase. Because ammonia is toxic, the urea cycle converts excess nitrogen into urea for safe excretion. I also included methionine metabolism in the formation of S-adenosylmethionine, which acts as an important methyl donor in the body. The pentose phosphate pathway branches from glycolysis and produces NADPH and ribose 5-phosphate. NADPH is important for reductive biosynthesis and antioxidant protection, while ribose 5-phosphate is required for nucleotide synthesis. Also included both purine and pyrimidine synthesis pathways. Purines are synthesized onto the ribose ring to form IMP, which can later become AMP or GMP. Pyrimidines are synthesized separately before attachment to ribose phosphate, eventually forming nucleotides like UTP and CTP. So overall, this poster demonstrates how interconnected metabolism really is. Pathways involving carbohydrates, lipids, amino acids, and nucleotides are constantly communicating through shared intermediates like pyruvate, acetyl-CoA, oxalacetate, and ribose 5-phosphate. The major goals of metabolism are generating ATP, maintaining energy balance, synthesizing important biomolecules, and supporting normal cellular function. So, really, creating this poster helped me better understand metabolism as one integrated network rather than a collection of separate pathways.